So whether it's pulse chase or GFP fusion protein, we can look at where breakdowns in intracellular trafficking happen by looking at mutants and what are they not able to do. And so here's a quick question. Given strain A, B, C, and D, and where you see the tag, which one's the wild type, and of the other three, what is the breakdown that we see? I'm going to let you pause here for a second and see if you can work it out, and then we'll go over the answer. So I'm guessing that you paused and you've got the answer. So let's talk about who's the wild type. Since the default pathway is secretion from the cell, strain B is the wild type. We see protein being made in the ER, being trafficked through the Golgi, and then moved into secretory vesicles, and then the protein in the extracellular space. So what's wrong with strain A? There's some fault in the movement of the protein from the ER to the Golgi. So it can't move the protein out of the ER. What's wrong with strain D is movement through the Golgi. The protein is getting accumulating in the cis side of the Golgi and not progressing through the Golgi. And the problem with strain C is the retention in the Golgi, so it's not moving from the Golgi to the secretory vesicles. And so so we can do experiments like this to find what the parts of the intracellular trafficking matter for which portion of the process. So so by getting a bunch of mutants, we can look at what they fail to do, and then just use genetics to find out what the mutation is, and then where that mutation is, we know that protein has something to do with that function. So we know that strain D, for instance, once we find the protein that has the mutation, we know that protein has something to do with progression through the Golgi. We know that strain C has something, the mutation has something to do with exit from the Golgi. So this is the hardcore cell biology process that allows us to use a genetic and tagged cell biology approach to finding mutants and therefore proteins that have to do with intracellular trafficking. So the next thing we want to know is how are things moved into vesicles? What is it about strain C that is not allowing for things to leave the Golgi and go into vesicles? So the default pathway is to bud off proteins into vesicles, sort them in the Golgi into where are they headed, whether they're going to the lysosome or they're going to a peroxisome or they're going to wherever they're going. The cell membrane or exocytosis from the cell. It's going to depend on whether the protein is correctly folded or not. So there are ways that a protein cannot be folded, like for instance heat shock. And if the protein is misfolded, it is going to be retained in the Golgi because we don't want misfolded proteins out doing harm to the cell. And so we have what were originally called heat shock proteins because that's how we discovered them. We heat shocked the cell, and then these were the proteins that were upregulated. Today we call a lot of them chaperone proteins, and chaperone proteins help proteins to fold in their correct conformation. And so how is it that these proteins get upregulated in the case of heat shock or any other thing that is going to lead to unfolded proteins? There is a signaling mechanism. So the protein gets made and moves into the ER lumen, and it either folds correctly or it doesn't. If it doesn't fold correctly, then a chaperone protein will help it to fold correctly. If the chaperone protein cannot, if there aren't enough chaperone proteins to help all of the misfolded proteins because of a heat shock or some chemical shock or something like that, then there will be a whole bunch of misfolded proteins. Those misfolded proteins will have hydrophobic amino acids exposed. And those hydrophobic amino acids exposed are a signal that is received by misfolded protein receptors. And so these receptors are then going to cause some sort of signaling cascade that ultimately leads to a transcription factor that gets imported into the nucleus. And remember, RAN, GTP, and the transporter proteins that help you to move into the nucleus. But the transcription factor is going to move into the nucleus and bind to the promoter for more chaperone proteins. And then those chaperone proteins will then, after transcription, then the mRNA will lead to more chaperone proteins being made, and those extra chaperone proteins will clear the misfolded protein. So this is called the misfolded protein response, and it upregulates these heat shock proteins or chaperone proteins in response to an excess of misfolded proteins. So then in the Golgi, we're also going to need some sort of tag that tells the protein where to go, because lots of proteins are in the endomembrane system, but they don't all go to the same location. Some of them get secreted from the cell, some of them end up in the plasma membrane, some of them end up in the lysosome. So some of them stay in the Golgi, some of them get transported back to the ER. And so what is the mechanism by which we recognize where the protein is going to be? And the answer to that is sugar. So as the protein gets made in the ER, a huge sugar gets added to it. And then it moves on to the Golgi. It first goes to the cis side and then progresses through the Golgi to the trans side. As these proteins mature through the Golgi, the sugar gets modified. We'll cut off some sugars, add some sugars. This process repeats until they have the correct sugar for wherever they're supposed to go. And that's all encoded in their own signal sequence. So if they have a retention in the ER signal sequence, then they will get the sugar that says retention to the ER. so they are tagged with sugars. So watch the video for progression through the Golgi, which is in the other PowerPoint slideshow. But let's just for now just recognize that there are a bunch of sugars. These sugars get modified, and that those sugars are then recognized by different receptors. These receptors bind to the sugar on one side, and then on the other side to specific transporter proteins. And so by modifying these sugars, we can package them into vesicles, and those vesicles will end up in the correct location of where they're supposed to be. So if they are a lysosomal protein, then they will get packaged into a vesicle that's headed for the lysosome, and then delivered into the lysosome. So so here's another good question. So in some lysosomal storage diseases, we see proteins that get stuck in the lysosome and don't get degraded, and we see the enzyme that normally modifies sugars. So it's a enzyme that adds phosphate to mannose 6-phosphate oligosaccharides in the Golgi. So that protein that adds these phosphates, we see that in the blood of people who have this lysosomal storage disease. So so the question is, what would be the mechanism for why we would see this enzyme that is normally in the Golgi in the blood instead, and how does that lead to these inclusions in the lysosome? So I'll let you pause the video for a second, and then we'll come back to the video and work through these answers. So I'm assuming that you did pause the video and worked out what your answer is, and the answer is C. Without the phosphate on the mannose 6-phosphate, they would not be targeted to the lysosome. And so the default pathway for this enzyme is to go outside of the cell. The secretion is the default pathway, and so everything that is supposed to be in the lysosome has to have a mannose 6-phosphate on it, and because the enzyme does not have mannose 6-phosphate on it, it gets secreted into the blood or secreted out of the cell, and so it's not in the lysosome where it would be breaking down those inclusions. And so I hope that that was obvious. Question A, or answer A, is pretty good. It says a target sequence, but they do have the correct signal sequence. It just doesn't have the mannose 6-phosphate added to it, and so so A is pretty good. The reason I don't like B is because the mannose without a phosphate is not the signal to be exported from the cell. export from the cell is the default pathway.